Hello everyone, I'm Zhang. Today I'm glad to share our recent work linear sampling and its findings on SARS-CoV-2. This work is done together with Liang Zhang, Sijun Li, Professor David Matthews, and Professor Liang Huang. We know that many RNAs fold into multiple structures. Partition function and base pair probabilities are widely used to compactly represent the exponentially large ensemble. But beyond these, we are often interested in generating alternative structures according to their Boltzmann probabilities, which can be provided by the sampling algorithm. One important application of sampling is to predict the accessible regions of RNA. Higher accessibility indicates that this region is easier for oligos to attach. From sample structures, we can calculate the accessibility by counting how many of the structures have the region of interest completely unpaired. However, the standard sampling algorithm suffers from three limitations. First, its formulation and implementation are unnecessarily complicated. Secondly, the sampling phase recomputes especially for large sample sizes. Finally, it is too slow to be used for long sequences, including the full genome of SARS-CoV-2. To address these issues, we developed a better sampling algorithm, linear sampling. It first simplifies sampling with a hypergraph framework. Using ECCB as a very simple example, during the partition function phase, RNA folding can be viewed as a framework of directed hypergraphs which are built in a bottom-up way. And the sampling phase is just to sample these hyperedges in a top-down way. We notice that some of the hyperedges are more important and revisited many times. So we store hyperedges at the first time that they are visited to avoid redundant work, which we call it a lazy-saving strategy. Finally, we linearize and speed up the classical sampling algorithm by beam pruning heuristics and successfully scale up to the full length of SARS-CoV-2 genome. Different from local folding algorithms, we don't have any limit of base pairing distance. For example, many of our sample structures have long interactions between 5' and 3' ends, which are typical for mRNAs. We compare the efficiency and scalability between linear sampling and Vienna RNA suburb, a widely used sampling tool on a dataset up to 30,000 nucleotide. In terms of end-to-end runtime, linear sampling runs in linear time and is orders of magnitude faster than RNA suburb. On Ebola virus sequence, which is about 19,000 nucleotide, linear sampling is 176 times faster. Also, for full length of SARS-CoV-2, linear sampling can finish it very fast while RNA suburb runs out of memory. In terms of sampling only time, linear sampling is 3 times faster on Ebola. Memory-wise, linear sampling only takes 1 gigabyte on Ebola, while RNA suburb takes 8 gigabytes. Next, we investigate the quality of samples. Our first metric is to evaluate if the sample structures approximate to the ensemble distribution. To do this, we calculate the average RMSD between the base pairing probability matrices derived from the sample structures and from Vienna RNA fold or linear partition. We can see that the average RMSD are quite small. For example, even with sample size 10, the RMSD between linear sampling and RNA fold is only 0.015 and is even smaller between linear sampling and linear partition. With the sample size increases, the average RMSD goes very close to 0. On the other hand, we want to know if the sampled structures correlate to the ground truth structures. So we calculate 2 kinds of defects. The first one is ensemble defect. which is the expected numbers of incorrectly predicted nucleotides over the ensembles. And the second one is accessibility defect, which measures how much the predicted accessibilities deviate from the ground truth. Lower defects indicate better correlations. We can see that linear sampling's ensemble defects are lower on most of the families, especially on the longer ones. Also, linear sampling overall accessibility defects are lower 523S RNAs. We also observed that the local mode of RNA suburb have higher accessibility defects, and surprisingly, it costs more time than its global mode. On SARS-CoV-2, linear sampling only takes about 69 seconds to sample 10,000 structures. We compare these structures to the canonical structures, such as 5' UTR canonical structure, and observe good correlations. From these 10,000 structures, we find 23 accessible regions of 15 nucleotides. Some of these regions are overlapped, resulting in a total of 9 separate accessible regions in 5 genes. These regions are potential targets for siRNA drugs and RT-PCR primers. Please refer to our preprint code and web server if you are interested. Thank you very much.